Good morning and a very warm welcome to all. Today we are going to discuss the important topic among the microbial metabolism, that is anaerobic respiration in bacteria. So anaerobic respiration, it is nothing but the respiration using electron acceptors other than molecular oxygen. In the case of aerobic respiration, we have already discussed that respiration using the molecular oxygen. Although the oxygen, it is not the final acceptor, the process still uses a respiratory electron transport chain. So in this anaerobic respiration, the oxygen is not a final electron acceptor. In aerobic organisms which are undergoing respiration, electrons are shuttled in electron transport chain, and the final electron acceptor is nothing but oxygen in air respiration. And the molecular oxygen, it is a high energy oxidizing agent, and therefore it is an excellent electron acceptor. In anaerobes, other than oxidizing substances such as nitrate, fumarate, sulfate or sulfur, so other than the molecular oxygen, these are the substances which are used as electron acceptor: nitrate, fumarate, sulfate as well as sulfur. And these terminal electron acceptors have smaller reduction potential than oxygen. So it have a smaller reduction potential comparing with oxygen, meaning that less energy is released per oxidized molecule. So if the molecule having smaller reduction potential means lesser energy are released. Therefore, anaerobic respiration, it is less efficient than aerobic. Why? Because less energy is released. That's the reason. And we can compare with the fermentation. It is also a type of anaerobic mechanism, but it is quite different from fermentation. So we can compare. Anaerobic cellular respiration and fermentation, both of them generate ATP but in a very different ways. And the terms should not be treated as synonyms. Cellular respiration, which both aerobic and anaerobic, it utilizes highly reduced chemical compounds. So the highly reduced chemical compounds such as NADH and FADH2. So these are the intermediates, and these are present in cellular respiration. For example, produced during glycolysis and TCA cycle in the case of aerobic respiration to establish, which helps in establishing an electrochemical gradient, and it is often a proton gradient across a membrane. This may result in what? An electrical potential or ion concentration difference across the membrane. Electrical potential means there is a difference in the concentration of ions across the membrane. So the reduced chemical compounds are oxidized by a series of respiratory integral membrane proteins, which I have discussed in electron transport chain, with sequentially increasing reduction potentials. And the final electron acceptor is nothing but oxygen in the case of aerobic respiration, and other chemical substance in the case of anaerobic respiration. So they are different from fermentation. A proton motive force drives proton down the gradient across the membrane through the proton channel of ATP synthase. So there is in the electron transport chain, there is a proton channel called ATP synthase, and the proton motive force drives the proton down the gradient through this proton channel. The resulting current drives ATP synthesis from the ADP and inorganic phosphate. So what happens as a result of the movement of this proton motive force, which drives the proton down the gradient, an ATP molecule is synthesized from ADP. But in the case of the fermentation, this process doesn't take place in fermentation. It does not utilize an electrochemical gradient. In the case of aerobic as well as anaerobic respiration, utilization of electrochemical gradient takes place, but in fermentation it doesn't take place. Fermentation instead only uses what? Substrate level phosphorylation to produce ATP. So we can discuss later about the substrate level as well as oxidative phosphorylation. I have mentioned in different. In the glycolysis I have mentioned, TCA cycle I have mentioned, electron transport chain also I have mentioned. So we can separately study in another video, that is substrate level phosphorylation, because it is an important question. Okay. So fermentation utilizes substrate level phosphorylation for the production of ATP. The electron acceptor NAD+ is regenerated from NADH formed in oxidative steps of the fermentation pathway by the reduction of oxidized compounds. So the NAD+ is regenerated from NADH, which are formed in the oxidative steps of the fermentation pathway, and this may take place because of as a result of the reduction of oxidized compounds. These oxidized compounds are often formed during the fermentation pathway itself, but may also be external. For example, while we are in the later sections, we can discuss about fermentation. In the case of the homofermentative lactic acid bacteria, this NADH is formed during the oxidation of what? Glyceraldehyde 3-phosphate. And it is oxidized back to NAD+. How? By the reduction of pyruvate to lactic acid. And this may occur at a later stage in this pathway. In the case of the yeast, acetaldehyde is also reduced to ethanol to regenerate what? NAD+. So NAD+ can be regenerated in the yeast by the reduction of acetaldehyde to ethanol. Now there are two important anaerobic microbial methane formation pathways through how? Carbon dioxide or bicarbonate reduction or acetate fermentation. So these are the anaerobic microbial methane formation pathways, okay, coming under anaerobic respiration. Now what are the ecological importance of this anaerobic respiration? Anaerobic respiration, it is a critical component of the global nitrogen cycle, iron, sulfur and carbon cycles. How? Through the reduction of oxyanions of nitrogen, sulfur and carbon. Reduction of this anions of nitrogen, sulfur and carbon, and it may result in the formation of more reduced compounds. Next point, the biogeochemical cycling of these compounds, which depend upon anaerobic respiration, significantly impacts the carbon cycle and global warming. Okay, so this may result in have an impact on the carbon cycle and global warming. Third point, anaerobic respiration occurs in many environments, including freshwater, marine water, sediments, soil, subsurface aquifers, deep subsurface environment and biofilms also. This anaerobic respiration occurs. Even environments such as soil that contain oxygen also have microenvironments that lack oxygen due to the slow diffusion characteristics of oxygen gas. An example of ecological importance of anaerobic respiration, it is the use of nitrate as a terminal electron acceptor or dissimilatory denitrification, which is the main route by which nitrogen is returned to atmosphere as molecular nitrogen gas. So these are the ecological importance of anaerobic respiration. Another example is methanogenesis. It is a form of carbon dioxide respiration that is used to produce a gas called methane by anaerobic digestion. The biogenic methane, it is used as a sustainable alternative to fossil fuels. On the negative side, when the uncontrolled methanogenesis in landfill sites release what? Large volumes of methane into the atmosphere, which acts as a powerful greenhouse gas. Sulfate respiration produces hydrogen sulfide, which is responsible for the characteristic rotten egg smell of coastal wetlands. So, sulfate respiration results in the release of the hydrogen gas. It has a foul smell, rotten egg, and it has a capacity to precipitate heavy metal ions from the solution, leading to the deposition of sulfidic metal ores. Next, in anaerobic cellular respiration, the production of energy, it requires oxygen. The points which I have discussed, I am again sum upping. So, the production of energy requires oxygen. That we already know. The electron transport chain, where the majority of ATP is formed, requires a large input of oxygen. So, for the taking place in electron transport chain, it requires a large input of oxygen. In many organisms have developed strategies to carry out metabolism without oxygen, or can switch from aerobic to anaerobic cell respiration when the oxygen is scarce. So what happens during cellular respiration? Some living systems use an organic molecule as the final electron acceptor. The processes that use an organic molecule to regenerate NAD+ from NADH, they are collectively known as fermentation. Certain prokaryotes, including some species of bacteria, some species of archaea, they may use anaerobic respiration. For example, the group of archaea called Methanogenesis, they have already discussed, reduced carbon dioxide to methane. For what? To oxidize NADH. And these microorganisms are found in soil and in the digestive tracts of ruminants such as cows and sheep. Similarly, sulfate reducing bacteria and archaea, most of which are anaerobic, they reduce sulfate to hydrogen sulfide to regenerate NAD+ from NADH. Eukaryotes can also undergo anaerobic respiration. Some examples include what? Alcohol fermentation in yeast and lactic acid fermentation in mammals. Now, what are the economic relevance? The dissimilatory nitrification. It is widely used in what? Removal of nitrate and nitrite from the municipal wastewater, and sometimes an excess of nitrate lead to eutrophication of waterways, in which the treated water is released. Eutrophication means the water cannot be used for the drinking or any other purpose. Full green color due to the development over production of algae, etc. Elevated nitrite levels in drinking water can lead to problems due to its toxicity. So it is highly toxic. Denitrification converts both compounds into harmless nitrogen gas. Specific type of anaerobic respiration are of critical in what? Bioremediation, which uses microorganisms to convert toxic chemicals into less harmful molecules to clean up contaminated beaches, aquifers, lakes and oceans. So this method of bioremediation is used. Then, for example, toxic arsenate or selenate can be reduced to less toxic compounds by various anaerobic bacteria. So this arsenate or selenate are highly toxic. They can be converted to less toxic form by using various anaerobic bacteria through anaerobic respiration. And the reduction of chlorinated chemical pollutants such as vinyl chloride and carbon tetrachloride are also occurs through anaerobic respiration. Then anaerobic respiration is also useful in generating what? Electricity in microbial fuel cells. which employ the bacteria that respire soil